everyone. Welcome to Bio Universe. I am Su Yanka and today we will discuss Golgi apparatus. Before that, please subscribe my channel. Golgi apparatus is single membrane cell organelles. It is a cytoplasmic organelle of smooth membrane sacs or cisternae, tubules and vesicles which takes part in elaboration and secretion of complex biochemicals. Synonyms of Golgi apparatus. It is also known as Golgi complex because it is made up of series of stacked cisternae. Dalton complex is the ultrastructure of Golgi apparatus as given by Dalton. Lipochondria due to temporary storage of lipid vacuole. The independent subunits of Golgi bodies are called dictyosomes, especially in plants. A liver cell may have 50 dictyosomes called Golgisomes. In fungi, Golgi bodies is called ancestral dictyosomes. Golgi complex is surrounded by a zone of cytoplasm which devoid of ribosomes, mitochondria, chloroplasts, storage granules, etc. It is called zone of exclusion. In 1867, George was first who seen Golgi apparatus. In 1898, Camillo Golgi was able to study the same in nerve cells of barn owl and cat by metallic impregnation method. He termed it as apparato reticolare or interno. In 1954, Dalton and Felix studied the detailed structure of Golgi apparatus. Occurrence. It is found in all eukaryotic cells except mammalian RBC, sieve tube elements, gamete cells of bryophytes and pteridophytes prokaryotes. Structure of Golgi apparatus. A compact Golgi apparatus possesses 4 types of components: cisternae, tubules, vesicles and Golgi vacuoles. Cisternae of Golgi are interconnected, stacked together, form called Golgi stacks. Each Golgi stack has 2 distinct faces, that is CIS face and trans face. CIS face is a proximal convex formative face, and trans face is a distal concave maturation face. Thickness of membranes of cisternae increases from forming phase towards maturing phase. Light thickness of first cisterna is 50 to 60 angstrom. Thickness of second cisterna is 60 to 65 angstrom, thickness of third cisterna is 65 to 70 angstrom and fourth cisterna is 75 to 80 angstrom. The phenomenon of passage of membrane components from proximal end to distal end of Golgi complex is called membrane flow. Tubules, they are short, branched, interconnected hollow filamentous substructures. It develop on maturing phase of cisternae. They are 30 to 50 nanometers in diameter. They are specifically involved in elaboration of secretory products. Vesicles, they are small sacs which develop from the tubules. They are pinched off as bud from forming face of proximal cisterna. They are of 2 types, smooth and coated. Smooth vesicles contain secretory products, also called secretory vesicles. They are of 20 to 80 nanometers in diameter. Coated vesicles covered by fine bristle-like outgrowths. They are 50 nanometers in size. Golgi vacuoles, they are large rounded vesicles or sacs which are produced by the concave maturing phase of distal cisterna. They have amorphous or granular contents. Example: lysosomes. Functions of Golgi complex. 1. It is complexing of lipids and proteins with carbohydrates. It produces glycolipids and glycoproteins. 2. Carbohydrates of cell wall matrix are synthesized in Golgi complex. 3. Golgi apparatus takes part in synthesis of special carbohydrates like galactose and sialic acid. 4. Hormones synthesis of endocrine glands is mediated through Golgi complex. 5. In oocytes, yolk is deposited around Golgi apparatus. 6. Fatty acids and glycerol absorbed by epithelial cells of small intestine are changed into lipids by smooth endoplasmic reticulum passed in Golgi apparatus, which packages the same into secretion vesicles. 7. In plants, cell plate is formed in middle of dividing cell through fusion of vesicles produced by Golgi apparatus. 8. Golgi complex forms acrosome, which is a special structure present near the tip of sperm that contains hydrolytic enzymes for digesting the protective coverings of egg. 9. Golgi apparatus receives membranes from smooth endoplasmic reticulum and transforms the same into coated vesicles. 10. They forms lysosomes.